Yesterday, news coaster was saying that Lever Davis has been changing titles all along. So I always have this, also have the same experience. So I make up my own title instead of following title. So the question is very broad. Can cell phone use cause cancer? So basically what it's saying is that what are the genetic effects of radio frequency fields that are emitted from cell phones? Now, to start with, this is a 10-minute talk, so I kind of simplify everything. If you have questions, just you can ask me specific questions. You can ask me after the talk. And now the first thing that I have to say is that we are not dealing with one entity, radio frequency field. It is a very complex entity that has different qualities. For example, the frequency, duration of exposure, the waveforms, those are the built-in quality of the exposure that determines the effects of the exposure. So when people tell you that, well, radio frequency field causes this, it does not generalize to all radio frequency fields. You have to ask, what are the frequency? What are the duration of exposure? What kind of modulation that you use in your exposure? Oh, my brain is here. Okay. Now, well, we have to talk about DNA, and fortunately or unfortunately, most of my talk has been given this morning. So genetic. The material, genetic material is DNA, deoxyribonucleic acid. So one conclusion is that radio frequency fields can damage DNA and cause, of course, when you damage DNA, the cell tries to repair, and in that process, it makes mistake, and it causes genetic mutation. And of course, genetic mutation can possibly lead to development of cancer. So this is a DNA schematic drawing of DNA molecule, which is also called a double helix, which is basically like two spaghetti twisted together. And they are joined by this so-called bases. Martin Planck was talking about this GA, those are these bases. So these two strains, these are DNA strains, the two strains of DNA molecule. And once in a while, bricks can form on the strain. Now, if a brick is formed on one of the strains, it's called single strain bricks. If very close by, on the other strain, there's another brick, the whole molecule will separate because they're so close together. If a brick form here, a brick form here, the molecule will separate. And that will form something called double-strand break. Now, double-strand break is a big problem because the cell lost the information of how to repair the DNA. And that is the time that mutation can happen. A single-strand break is not that serious because the other information is on the other side of the molecule. Now, in the RF radio frequency study, most of the DNA study use something called the Comet assay, which is a very sensitive method for doing DNA damage. You can measure DNA damage within one cell. And it's possible to do single-strand break, measure the single-strand break, and also measure double-strand break. Now, while we're talking about spaghetti, if you have a bowl of spaghetti, and then you put a pair of scissors into the bowl and chop, and the more you chop, the more small pieces will be formed, right? So now, if there's a possible that you pour all this spaghetti out and count how many of these small fragments come out, the more fragment, the more damage, right? So this is basically the principle of the common assay, is that we isolate the cell from, for example, from the brain, and then we take the DNA from that particular cell, and then we pull the DNA out from this DNA ball. And by doing that, the small fragments will come out. The more fragment you see, the more the damage, because that breaks form on the strain. And now this is a brain cell of a rat, isolated from the brain of a rat. This is a DNA ball that forms. There's some fragment coming out because the DNA is constantly being attacked and break, and of course the cell can repair. Now this is a cell from a brain cell without radiation, so you can basically form a very spherical ball. Now, this is two cells, two DNA balls. After exposure to radiofrequency radiation for 2 hours. And you can see the fragment coming out from the ball. The more fragment, the more damage, right? So let's go back. All right, it takes time. So this is without radiation. This is with radiation. So you can actually count the, you can use a computer program to count how many fragments come out from this DNA ball, and then you will see then it gives you some kind of quantitative measurement of the effect. Here are a list of publications that have been showing DNA damage after exposure to radiofrequency radiation from cell phones, mobile phone radiation. If you go into the literature of microwave radiation, the general spectrum, you will see more of these publications. And so each of this is one publication, a peer-reviewed publication. Now, what causes the damage? Radiofrequency field damage, DNA damage. One thing that I think is a very promising explanation is free radical formation from cells. Every cell that uses oxygen will form free radicals. And well, my students usually ask me what are free radicals. So they're very active molecules. Now, if imagine you have a ballroom and people are dancing inside the ballroom, and suddenly Fred Astaire comes into the room and grabs the lady and start dancing. Now, how many ladies can dance with Fred Astaire? So the whole ballroom will go into chaos. So that is the free radical. Okay. Now, so we know that this. How do you counteract free radical? We take antioxidants. For example, vitamin C, vitamin E, the antioxidant. So antioxidants are actually is a Ginger Rogers. So if you put it into a ballroom, and eventually Ginger Rogers will dance with Fred Astaire, so the whole ballroom will go, huh? Oh, of course. So Dr. Haberman was talking about this, this using antioxidant to study the effect of free radicals.